What I'd like to do is to go back to a little bit of basic science, and I'll talk to you about some of the work that we've been doing over the last few years that deal with a non-pharmacological method to perhaps approach acute kidney injury. So this is the outline. It will go over the inflammatory reflex to control systemic inflammation and methods to activate the inflammatory reflex to attenuate acute kidney injury. There's going to be electrode-based discussion, ultrasound, and some of the newer areas, which includes optogenetics. Now, the nervous system and the immune system are at the heart of this talk, and both systems were identified and they began to be studied around 1900. The immune system was championed by Metchnikoff and Ehrlich, who pioneered the innate and adaptive immune systems, respectively, and they were jointly awarded the Nobel Prize in 1908. Now, the nervous system was pretty much discovered by Santiago Ramon Cajal. And I put this picture up here because just 2 weeks ago I was in Barcelona, and we were sauntering down the old city, and my wife noticed this sign here, which was the place where Dr. Cajal was doing his research. And he really was, and my wife is a neuroscientist, and immediately she recognized that this was the person who was referred to as the father of modern neuroscience. Now, both of these systems have been studied autonomously for many, many years, up until about 25 years ago. They were thought to be independent, and they were studied as if they were separate systems. But about 25 years ago, Linda Watkins made the seminal observation that linked these two systems together. And what she observed was that if you inject IL-1 beta into the peritoneum, this causes systemic febrile response. And it was thought at that time that perhaps maybe the IL-1 beta got into the circulation and passed through the blood-brain barrier to cause the febrile response. But it turns out that it was something very different. She cut the vagus nerve, and it turned out that once she cut the vagus nerve in the abdomen, that this prevented the febrile response. So it turns out that that IL-1 beta binds to the toll-like receptor, or binds to its receptors in the sensory afferent neuron of the vagus, which sends signals to the brain, which cause the febrile response. But this seminal finding then led to studies which then determined that there is a close interaction between the nervous system and the immune system. Now, one way that this could be very important is in sepsis. For instance, if you have a septic response, there's a huge hyperinflammatory response. And the immediate response is that there's an immunosuppressive response, or a response that suppresses this inflammatory response. And if it's equal and opposite, then it's balanced, and the human could probably recover. However, if it's unbalanced, for instance, if there is no adaptive response, there's overwhelming inflammation and death. Now, the cholinergic anti-inflammatory pathway is described here. And essentially what this shows is that there's the inflammatory cytokine release, whether it's TNF or IL-1 or whether it's PAMPs or DAMPs-like molecules. It binds to the receptors on the afferent vagus, leading to the dorsal motor nucleus of the brain. This then sends a reflex pathway to shut down the inflammation through the efferent vagus, which leads to the heart, which causes heart rate variability, and in neonates, this is the first sign of sepsis. It also goes to the visceral organs, one including the spleen, in which it binds to the alpha-7 nicotinic acid receptor on macrophages to then decrease TNF-alpha release, and it causes this anti-inflammatory response to sepsis. If you look at the inflammatory reflex a little bit more carefully, you can see that it terminates on the celiac ganglion. It then synapses with the adrenergic splenic neuron. This then releases catecholamines to bind to the beta-2 receptors on the T cells, and these are very special T cells, which I'll get into in a few minutes, releasing acetylcholine, which binds to the alpha-7 nicotinic acid receptor on macrophages. This then blocks the release of cytokines. Now, what happens is that the alpha-7 nicotinic acid receptor is activated, it blocks NF-kappa B translocation into the nucleus, and this decreases the cytokine response. These T cells are very important in that they're different kind of T cells. They are choline acetyltransferase containing T cells. They're CD4 positive, CD62 ligand positive cells which secrete acetylcholine, unlike other T cells. Now, if you go into the spleen, you can see that in the white pulp, you can see all the CD3 cells which are in red, and you can see these chat-positive CD3 T cells, which are shown here. And if you look closely, you can see that these green cells, which are the chat-positive T cells, you can see that the synaptic nerve terminals synapse very closely next to the T cells. And functionally, if you look at the red pulp, you can see all the macrophages which contain tumor necrosis factor, shown in red. And if you stimulate the vagus nerve, you can see that it decreases the production of TNF-alpha, and you can see that, that there is no TNF-alpha production in the macrophages. Now, the cholinergic anti-inflammatory pathway has been described in a number of different animal models of colitis, sepsis, arthritis, pancreatitis, and others. In fact, there are many clinical trials now that's looking at the use of ways to block or use ways to activate the cholinergic pathway. One potential important situation would be in brain death, in organ transplantation. We know that when there's brain death, there is a huge release of neuropeptides, catecholamines. There's a lot of metabolic derangement. And if you stimulate the vagus nerve in an animal model and transplant the kidney after brain death, you can see that the histology is much better preserved in those animals that receive vagus nerve stimulation compared to those animals that not receive vagus nerve stimulation. And if you look at functionally, 16 weeks later, you can see that those animals that receive vagus nerve before transplantation and before brain death, you can see that their plasma creatinine was lower than in those animals that did not receive vagus nerve stimulation. So there is a potential real importance in terms of the role of the cholinergic anti-inflammatory pathway. Now, in humans, there are a couple of clinical trials, and early preliminary studies are shown here. This is the cholinergic vagus nerve stimulation in Crohn's disease by Bonaz, and there is one in terms of rheumatoid arthritis in the group by Kevin Tracy. And so you can see that there are clinical trials in which they're targeting the vagus nerve for inflammatory conditions, and in these studies there are very few side effects from these studies, and it has shown to be effective. Now, what we wanted to do was to look to see if the vagus nerve pathway can be important in terms of controlling inflammation and acute kidney injury. So we took black 6 mice, we stimulated the left vagus nerve with 50 microamps, 5 hertz for 10 minutes, and then this was 24 hours before ischemia reperfusion. 24 hours later, we then euthanized the animal, and we obtained blood, kidney, and spleen. And these are the results. What you see is plasma creatinine on the vertical axis in the various conditions. If you take the animal and you perform ischemia perfusion, the plasma creatinine rises up to about 1.5 milligrams per deciliter. If you perform vagus nerve stimulation on the intact vagus nerve 24 hours before IRI, you can see the plasma creatinine is markedly reduced. If you transect the nerve and stimulate the afferent end going towards the brain, you can see it's equally effective. And if you transect the nerve and stimulate the efferent end, you can see that it's also very protective. If you look at the kidney injury molecule 1, you can see very similar results. That is, in all 3 situations, vagus nerve stimulation are protected. And I don't have it here, but the histology also shows protection as well. Now, this is a summary of the results of the study. And what this shows is that if you cut the left vagus nerve, stimulate the afferent end, it causes marked protection. If you stimulate the efferent end, it also causes marked protection. If you stimulate the afferent end and anesthetize the right vagus so that there's no transmission, it's also protective, suggesting that there's probably an alternate pathway as well that leads to a protective effect. Furthermore, this protective effect of vagus nerve stimulation required CD4 T cells. It required the nicotinic acid receptor on bone marrow cells and on macrophages. It required the beta-2 receptors. And also, if you transfer the splenocytes from vagus nerve stimulated animals, it also transfers protection. So this is the diagram which illustrates that experiment, that if you take donor mice and you stimulate the vagus nerve versus sham stimulation, you then isolate the splenocytes and you transfer them into recipient mice, perform ischemia reperfusion on the recipient mice. These animals that received vagus nerve stimulated donor cells were protected as opposed to those that received the sham stimulated cells. So this indicates that something about vagus nerve stimulation conditioned these splenocytes that led to the protection in the recipient mice. Now, the clinical use of vagus nerve stimulation are quite varied, and you can see that it's approved for epilepsy and depression, but there are clinical studies looking at all these different conditions in terms of vagus nerve stimulation. However, what we do know is that there are no studies in kidney disease. Now, vagus nerve stimulation is invasive. And what I want to talk about over the next couple of minutes is that we found that ultrasound, pulsed ultrasound, has a very similar effect to vagus nerve stimulation and provided the same degree of protection, but it's a non-invasive approach to the same issue. What we did was we took the animals and we localized the kidney or the spleen using grayscale mode. We then administered 20 pulses of ultrasound and with a mechanical index of about 1.2. This is done 24 hours before ischemia reperfusion. And what you'll observe is that the ultrasound induced a marked protective effect compared to the non-ultrasound treated animals. If you perform the ultrasound 1 day before, you can see that there is marked protection. If you perform the ultrasound 2 days before, you can see there's a marked protective effect. And then if it's 3, 5, or 7 days, you can see the protective effect was lost. If you look at the kidneys 4 weeks later, there is less fibrosis compared to the kidneys that did not have ultrasound. If you look at the fibrotic markers shown in the ultrasound treated, which is shown in the white bars, you can see that the alpha smooth muscle actin, collagen, vimentin were markedly reduced compared to those animals that did not receive ultrasound treatment. Furthermore, if you look at mortality, you can see that those animals that received ultrasound, that they lived to a much higher percentage than those animals that did not receive ultrasound treatment. So this indicated that prior ultrasound treatment reduced ischemia reperfusion injury, fibrosis, and death. Now, we've gone through a series of experiments which really delineated that the cholinergic anti-inflammatory pathway was at the heart of this. We know that ultrasound treatment protected the animals from ischemia reperfusion injury. If you denervate the spleen, the protective effect of ultrasound was lost. If you use a beta-2 antagonist, the protective effect was lost. If you use bungarotoxin, which is an alpha-7 nicotinic acid receptor antagonist, this also blocked the effect of ultrasound. If you used a cholinergic agonist, this mimicked the effect of ultrasound. All these studies suggest that the cholinergic anti-inflammatory pathway mediated the protective effect of pulsed ultrasound treatment. In addition, just like in vagus nerve stimulation, if we performed ultrasound on donor mice versus no ultrasound, and we transferred the splenocytes into recipient mice, the animals that received splenocytes from the ultrasound-treated animal were protected as opposed to those that were from the no ultrasound-treated animals. And that data is shown below. And you can see that these animals were subjected to ischemia reperfusion injury. They received splenocytes that were from the animals that had no ultrasound treatment. The plasma creatinine was very high. If you use 2 × 10⁵ splenocytes from the ultrasound-treated animal, you can see some degree of protection. If you used more splenocytes, you can see there was even more protection. So again, the ultrasound somehow conditioned the splenocytes that led to the protective effect. So in order to move this further, what we wanted to do, and in order to dissect the neural pathways, was to use a technique called optogenetics. And my colleague, Patrice Guillenet, is an expert at this, and he's been helping us with these studies. And optogenetics involves exogenous genes that code for light-sensitive proteins, and they're expressed in specific cells that upon exposure to light, activate or inhibit the nerve. So if you use channel rhodopsin, this activates the nerve. And so you can introduce channel rhodopsin into specific nerves and introduce blue light. This then, we hope, would decrease ischemia reperfusion injury. The advantage of this is that you can see that if you could use specific genetic approaches, you can introduce channel rhodopsin only in certain neurons, but not in other neurons. And so I show this pathway because this links the central nervous system to the peripheral nervous system. The vagus has afferent and efferent limbs. And you can see there's these different regions within the brain. One of the regions is the C1 neuron, which connects with this pathway. And we use the C1 neuron first to be able to detect the effects of optogenetic stimulation. And we know that these C1 neurons are activated by a variety of stresses, such as infection, hypotension, and presumably ischemia reperfusion. They contribute to autonomic and neuroendocrine responses. And in vivo, photostimulation of the C1 neurons activates the efferent vagus nerve. So we injected the virus 5 to 6 weeks ahead of time in order to introduce channel rhodopsin into the C1 neurons. We then performed stimulation with 5 hertz and for 10 minutes for 24 hours before ischemia reperfusion. And then 24 hours later, we sacrificed the animals and obtained blood and kidney tissue. And these are the results. So if you perform ischemia reperfusion, you can see that the animals the plasma creatinine rises considerably. But if you perform ischemia reperfusion in those animals that received the channel rhodopsin in the C1 neurons, and you expose it to the blue light, you can see they're markedly protected. But if you use a reporter without the vector shine the light, you can see that there's no protection. This indicates that stimulating channel rhodopsin in the C1 neurons activated the vagal pathway, which then protected the animals from ischemia reperfusion injury. Now, it's important to realize that the vagus nerve is quite heterogeneous. There are nerve bundles that go to different regions of the brain, and there's the afferent and efferent pathway. And this one paper in Cell shows that if you introduce channel rhodopsin into specific neurons, you can get a different response. So we're interested in learning more about the heterogeneity of the vagus nerve so that we can provide more specific approaches. So we use the Krelox technique in order to introduce channel rhodopsin into the vagus nerve. And so V-GLUT basically is expressed in the afferent vagus nerve. And we know that 80% of the vagus nerve is afferent. And then we use CHAT-CRE to introduce channel rhodopsin into the efferent limb. And we only have preliminary data for the afferent, and it's very preliminary. But if you stimulate with the light, you would expect to see a response in terms of changes in ventilation and respiratory rate. And this movie here, I think, if I can get this to work. So the mouth is breathing, then we stimulate with blue light, and you can see the respiratory pattern change considerably. This is with 50 megahertz, so it's a very potent laser stimulation. In our ischemia reperfusion model, we only use 5 hertz. So if you look at the tracings, you can see that with light stimulation, you can see a ventilation stop, and this is with the 50 hertz, and we're going to use 5 hertz for our studies. And these are the preliminary results. Not too many dots, but you can see that they're, with laser stimulation, stimulating the afferent pathway, you can see there's a marked protective effect with this approach. Lastly, what we're trying to do is to be able to utilize both imaging and therapeutic pulses at the same time. And the problem is that with the current technology, you don't have significant localization. You don't know where the ultrasound is targeting. What we try to do with a grant that we submitted and we got funding for, is to develop a programmable ultrasound scanner. And this allows for both imaging and therapeutic pulses delivered at the same time. So we can image and deliver therapy at the same time. And you can see the problem is that the waveforms are very different for therapy and imaging. And in the past, using a conventional scanner, you have to do one or the other, but not both. In our preliminary study, you can see that with our new scanner, if you increase the mechanical index, there's a threshold at which you see a marked protective effect in terms of plasma creatinine, below which you can see that the plasma creatinine is very high and it's unprotected. So in conclusion, neural circuits that control immunity and inflammation offer novel targets in the kidney therapeutic space that may lead to the development of the next generation therapies to preserve organ function. The cholinergic anti-inflammatory pathway consists of an immune reflex pathway that can be activated by vagal nerve stimulation to block splenic-dependent systemic inflammation and acute kidney injury. Non-invasive, non-pharmacological approaches to activate the cholinergic anti-inflammatory pathway include ultrasound application and transcutaneous electrical stimulation of the vagus nerve. And lastly, optogenetic tools and techniques provide new opportunity to dissect the neural pathways that control immunity and inflammation and could provide novel approaches to therapies for kidney disease in the future. Thank you very much for your attention. And this is the individuals that participated in the studies, and they're shown highlighted in orange color. And I particularly wanted to highlight Tsuyoshi Inoue and Chikata Abe, John Hasek, and Patrice Guianet. And so thank you very much for your attention.